All right, welcome to Ideas on Tap, presented by the Museum of Natural and Cultural History. My name is Lauren, and I'd like to start this evening by recognizing that the museum and the University of Oregon are located on Kalapuya Ilihi, the traditional indigenous homeland of the Kalapuya people. Following treaties between 1851 and 1855, Kalapuya people were dispossessed of their indigenous homeland by the United States government and forcibly removed to the Coast Reservation in Western Oregon. Today, Kalapuya descendants are primarily citizens of the Confederated Tribes of Grand Ronde and the Confederated Tribes of Siletz Indians, and they continue to make important contributions to their communities, to the museum and to the university, to Oregon, and to the world. We express our respect to the many more tribes who have ancestral connections to this territory, as well as to all other displaced Indigenous peoples who call Oregon home. To everyone joining us this evening, welcome. Although we might not be together in person, we do still want to hear your questions and your comments. During this evening's program, you can use the Q&A and chat features on Zoom and Facebook to reflect on the talk or add your thoughts, to engage with other viewers, and to ask questions of our speaker. I'll be collecting your questions across both platforms, and we'll get to as many as we can at the end. During the talk, I'll post a link to a brief survey in the comments on both Zoom and Facebook. Please consider taking a minute or two to fill it out, even if you've already filled it out before. Your feedback will help us in future virtual programs, and you'll be entered to win a $25 gift card to Viking Braggot Company, where the museum hosted our Ideas on Tap program prior to the pandemic, and where we hope to be back soon. If you enjoyed tonight's program, we are asking that you please consider making a secure, tax-deductible donation of $5 to $10 at the web address listed on the screen to help keep museum programs like this one accessible to all. That's giving.uoregon.edu/MNCHgift. Your donation directly supports the museum's educational programming, bringing science and culture adventures to Oregonians of every age and in every corner of the state. Thank you. Before we get started, I have a couple of upcoming museum programs to share with you. The museum's newest exhibit, Photo Arc, opens this coming weekend. You'll come face to face with charismatic animals through stunning photography by Joel Sartor. During grand opening week, which runs from Saturday, May 5th to Sunday, March 13th, we'll have additional animal-inspired activities for all ages, both indoors and out. On Friday evening, March 4th, museum members are invited to a special members-only preview and celebration. If you're not yet a member, it's a great time to join. Visit our website to learn more. The museum's exhibits are open Wednesday through Sunday from 10 a.m. and until 5 p.m. and until 8 p.m. on Thursdays. The museum is currently inviting nominations for the 2022 Oregon Stewardship Award. The award recognizes an environmental or cultural heritage project that involves the community and aligns with the museum's mission to inspire stewardship of our past, present, and future. Nominations are open through March 13th. Visit our website to learn more or to nominate a project. This year has flown by, and after tonight, we have just 2 more Ideas on Tap remaining until we take a break for the summer months. I hope you'll join us in April for When a Solution is Not the Solution. A poison elixir may bring to mind images of witches and cauldrons, but what about well-meaning pharmacists? Join UO chemist Lindsay Hinkle for a discussion of how science helps ensure that good intentions don't have dire consequences. And then in May, we'll explore how the power of story can help us reckon with our past and shape our future. Join Kimberly Johnson, author of This Is My America and Vice Provost in the Division of Graduate Education and Student Success at the University of Oregon, for a discussion on literary activism and expanding silenced voices. We hope to see you there. Finally, since tonight's talk is about pheromones, or nature's love potions, if you will, I want to remind everyone to come and check out another of the museum's recent exhibits, Magic in Medieval Europe. Through spellbinding stories and objects, the exhibit delves deep into the phenomenon of magical thinking, past and present. Come and visit us. All right, and now it's my pleasure to introduce this evening's speaker, Dr. Robert Mason. Bob is a professor of integrative biology at Oregon State University. He is interested in the interrelationships among natural products chemistry, behavioral biology, reproductive endocrinology, and ecology. He conducts research both in the laboratory and in the field to uncover the role of hormones and pheromones in reproductive behaviors and physiology, with an emphasis on reptiles because they rely more on their chemical senses than any other vertebrate class. He joins us this evening for Snakes, Sex, and Sense, the Garden of Eden Revisited. Try saying that 3 times fast. Welcome, Bob.
Thank you, Lauren. That's a great, thanks for that kind introduction. Yes, that is a tough one there. I did go to a Jesuit college, so always the Garden of Eden always has lots of interesting things, but really it's more about the snakes and the sex part, which of course we all know that's what it was really about. Actually, I shouldn't say that. A lot of times I say that and people didn't know that's what it was really about. But it's I wish I were there in person like everybody else, and just like Lauren said, I hope we're going to get back to that very quickly. But let me just switch right to be a bad professor and switch right to my slides right away. Maybe there. And let me move myself up here. Whoops, get out of the way there. So yeah, there's our title, and there's a picture in the background. So I will give my little standard warning. If you really are have a terrible phobia of snakes, this may not be the best talk to be in, because I'm going to show you lots of snakes and videos and all kinds of things. But even if I usually find that either people are really fascinated by it, even if they're horrified, they're just they're like, wow, I still think that's pretty cool. So they stay. All right, so let's see. Here, I was going to say already I can't get it. Lauren gave a good introduction to what we do. One of the things, it's kind of a mouthful and sounds like a hodgepodge, but it's one of the things I think is really great about biology, and even more so now. I'm getting to be long in the tooth. But you can really, the days of having somebody that just wore one hat, I'm a comparative anatomist, or I'm a mammalian physiologist, or I'm a digestive endocrinologist, like those days are gone. Now, people really wear lots of different hats, and I have to admit, it's really a lot of fun. And so people do often think, well, how can you be doing chemistry and field work way up in remote parts of the world, and in pheromone biochemistry and behavior and all that? And well, you do, and it's great. So this little cartoon just shows some of the stuff that we do. Whoops, where's my little arrow? There it is. I don't know if you can see it, but in the middle here is reproduction. That's what that's kind of the glue that holds us together. Everything here is about reproduction. And then Lauren told you some of the stuff we do. We do behavioral endocrinology. That's how hormones affect behavior. And so in this case, these hormones are mostly gonadal hormones, so gonadal sex hormones. So things like, testosterone, estrogen, progesterone, things like that. Although lots of brain hormones now, and that comes over here to neurophysiology. I'll show you just a touch of this. It's kind of a teaser talk. Behavioral ecology is a lot. We study our behavior and we study in the field. So we're not like white mice people. We don't just, have them in a cage in the lab or something. Not that there's anything wrong with that, but we study right on the field. I'll show you that. And then this last balloon is chemical ecology, and what that is really well, I'll show you in the next slide. Let me not even say. So, whoops, come on. So if two animals, or excuse me, if an animal is communicating within itself, so in this case, it's like this cell is communicating to that cell. We call that kind of chemical communication endocrinology, and the chemical signal are hormones. So people know about that. But what if you have two individuals? So in this case, we got one snake and it's interacting with this other snake. And the chemical cue in that case for this communication, because it's between two individuals of the same species, is called pheromones. If we have something like this, is another kind of chemical communication. In this case, this snake sees this yummy frog, and the frog is sending prey, what they're called pheromones, to the snake, so the snake can detect the frog by its chemical cues. And so that's another kind of chemical communication. And then finally, there's a type that are called predator, instead of prey pheromones, they're predator pheromones. So in this case, this weasel or mink, whatever, is saying, it's saying through chemicals, I'm a predator of you. And then the snake says, yikes, predator. And then the green arrow shows, phew, it tries to get out of there. So lots of interesting things that nature does with chemical cues. And so then one question that even my parents used to ask me way back when I was in grad school, why study pheromones in snakes? What's up with that? Lauren kind of talked a little bit about that. Snakes really do have very highly developed vomeronasal, I'll come to that in a minute and later, and olfactory system. So they smell, whoa, what's going on there? Get out of there. I'm stealing my own thunder. And they, as she said, they really rely more on their chemosensory communication than any other vertebrates. So even though snakes can see well, they can't hear at all because they don't have ears, so they don't hear, but they see well, they smell well, and then they have this other sense that we'll talk about. Snake pheromones are very robust. We'll talk about those too. You can do a lot of chemistry to them, and they're not wimpy, so they fall apart and can't do anything. I claim the behavioral bioassays are robust, quantifiable, and unequivocal, but I'm going to let you folks be the judge of that. And then, as I often say, the exception often illuminates the rule. And then a big thing for biologists is this. When it comes to snakes, you can get big sample sizes. And so that's not a barrel of snakes that I just threw on the ground or whatever to make a nice shot. That's what it really looks like up here in central Manitoba. So see this red dot right up here? So I always have to show this stuff because I've found that Americans are very woefully lacking in knowledge about our northern neighbors and their provinces. So this is a huge province, Manitoba, and it's here, you can see above North Dakota and Minnesota. And so where we go is right up here in what's called the Inner Lake region. And this big lake right here, Lake Winnipeg, is actually bigger than two of the Great Lakes. So it's a really big lake. This picture down in the lower left, I used to be kind of mean and say that's Manitoba at the nice time of the year, early in the spring. And you can see it is actually spring, but because there's no snow on the ground, but it's quite desolate, and it shows the geology of the area. The place is flat as a pancake. I used to have a grad student who was a big time rock climber and stuff, and he just about died up there. He said there's no relief in this province anywhere. It's like the tallest place up there is like 10 meters. So the glaciers scraped it flat. And then you can see these rocks. These yellowish rocks are limestone. They give an idea of what the geological history is. And then these gray ones over here, those are glacial erratics. So that stone came from way up here in what's called the Canadian Shield area. It goes across North America like this. That's some of the oldest rock in the world. So the glaciers scraped that off and then dropped them on here. The limestone, all of the bedrock here, in fact, all through the middle of North America. So through Manitoba, through Saskatchewan, and even into Alberta, and you folks probably know about that if you've ever been, and all through the Midwest of us, way down, the Dakotas, Kansas, Nebraska, well down into Oklahoma and Texas. Is a limestone substrate, and that's because it's the remnants of the great inland sea, Lake Agassiz. And so, of course, that was there for millions of years, all the marine life, died and settled to the bottom and stuff, and then it fossilized into, well, not fossilized, it morphed into through geochemical process, it turned into limestone, into a mineral. And where there is limestone and water, so there's a lot of water up in Manitoba, both from the snow melt and then from surface water, you'll often get something like this, that this is called a sinkhole. So you've probably seen these too. And so this is where the water has scoured out and dissolved the limestone in underground, and then through the freeze-thaw cycles up in Manitoba, it cracks the roof, and then the rubble falls down, and it makes an area. Whoops! I keep hitting my progress button here. Anyway, it makes a big sinkhole like this, and what that does is, if it's below the frost line, deeper than the frost line, which up in Canada is over 3 meters, so you're about 10 feet down, you're solid ice until you go about 10 feet down, then you hit soil. And so if you can get beneath that area, that's a great place for you to hibernate. It's still cold, but it's not freezing, because the temperatures on the surface in Manitoba, let's see, it's March now, it's starting to warm up, and we do have global warming. But anyway, it can get to about 50 below zero there. And so it can get quite cold. So it's hard for anything to survive. And then in the spring of the year, what happens is, just like birds, the male birds come back first in the spring when we're right at that point right now here in Oregon. The males will come to the breeding grounds first. So the male birds will fly up, they stake out a territory, they start singing, things like that. They start to build a nest, then the females come and they tear that nest out and build a proper nest. We can also hear, we're kind of bereft in frogs, but we do have, at this time of the year, you hear our Pacific tree frogs calling. And so they'll do that. If you go anywhere else in North America, you can hear just a din of amphibians calling, and so they do that. Same thing, males will go into the ponds first and try to get good spaces and things. Well, in this case, the breeding grounds are just the surface of the den. So this is down in the bottom of a sinkhole, and the snakes go in underneath that, and you can see all this rubble on it. They go down and beneath all that rubble, and they get down below the frost line there. And so in the spring, they come out. So these are the males, and the males all come out. And these dens are about the size of the average person's living room. So they're big, but not terribly big. And there's this particular den, there's about 75,000 snakes there. And so you're literally sort of ankle to shin deep in snakes in the springtime. Springtime up there is May. Okay, so here we go. So these are red-sided garter snakes, harmless. They're the kissing cousins of our red-spotted garter snakes that many of you will see around the water, near the water, or our northwestern garter snakes, wandering garters. They're all kissing cousins. They basically are all stripy guys like this. So these are all males, and all these males, what do you think males do in the springtime? So they're looking and looking. What are they looking for? Ah, there she is. They're looking for an unmated female that's just emerging for the first time this year. And so that's her. And what will happen is the males kind of all emerge first. So you'll get literally tens of thousands of males. But the females, for reasons we still don't understand, they come up only a few at a time. So if you look at any moment in time, if you took a snapshot up in Manitoba, you would see maybe 10,000 males and about 10 females. So the females and the males, the sex ratio of the population is equal. There are just as many females as males. We know that from the fall. When we come back, we can do surveys. But in the spring, what's called the effective sex ratio can be as skewed as 1,000 to 1, 1,000 males to 1 female. So you can imagine what's coming up next. So then what happens is, as soon as that female comes out of the ground, she's set upon by males. And in this case, let me show you my arrow. Hopefully you can see. So there's the head of the female. You can see here's her body. See this bigger body all the way down here, all the way, down into here, all the way up like this, curling back, and there's the end of her tail right up there. And all of these other guys are full-grown males. So right off the bat, you say, okay, there is sexual dimorphism in this species. The females are much bigger than the males. That is true, but you'll also see the coloring may not look like it, but there is no color difference between the males and the females. They're also, I told you before, you can kind of see it in this guy's head here or this one here. There's no hole in their head, so they don't have ears. And snakes don't make sounds anyway, so the things like the calling birds and the calling frogs, they don't use that kind of thing. And so what I'm getting at here is, and let me show you this and then I'll get to it. What happens when the males discover a female is they do this. They do two things. They start to court the females by chin rubbing. And so they rub their chin like this in a jerky motion up towards the head. And what they're trying to do is get their tails way back here where the business end is for both the males and the females. And then somewhere, it varies in the amount of time, somewhere along the lines, even though there's all these males, there can be 10 to 100 males swarmed around one female. She calls the shots. And so when she decides it's time to mate, she'll lift her tail and open her cloaca, and then the males will where's my arrow there? Males will intromit one of their two hemipenes into the female's cloaca, and then they mate for about 15 minutes. The male transfers the sperm into the female, and then he leaves a copulatory plug, which serves to both keep the sperm from kind of falling back out, but it also serves as a really nice plug to keep other males from mating after you did. I call it a hemipene, is what its name is. A terrible name. It implies half a penis, but what it really means is we herp guys say snakes have something extra. They have two copulatory organs. They have a right hemipene and a left hemipene. And of course, everybody is probably thinking, you know, why do they have two and humans only have one, or mammals only have one? And the better question is, and the smart aleck answer is, why do humans and mammals only have one when their ancestors had two? We can come back to that in the questions if we need to, but anyway, so yes, so this, so how do the males find the female? And you're like, well, Bob, it's right there. Look at the female is way bigger than the males. It's like they just see that. But let me show you here. We're going to talk about some of this pheromone chemistry now. And this is what these guys do. I often tell my students, you know, the kind of science we do, and I would claim the best science is really almost simple-minded sounding science. So in this case, this little guy is saying, hey, Dad, which ones are the girls? And you can see the mating balls. Oh, I didn't say that yet. Those aggregations around a female are called mating balls because they actually can ball up like this and fall down a hill, like roll down a hill or whatever. Here's the guys looking back and forth. So how do they find this? So here's the thing. Okay, the females are bigger. There is a female in this picture, I promise you. Can anybody pick it out? I can't see you all, so I'll just jump in. See this? Can you see my, whoops, can you see my arrow? This is the female right here. So I would claim it's the same thing for a male. If you're a male and you're on the ground, remember, you're not looking down from on high like we are, like that picture I showed you. What you see is, if you can see my face, is you're basically like kind of poking your way through a sea of living spaghetti there. And so that is, so how do you do that? So visual cues, I would claim, are out. Auditory cues, I told you, they're gone. They don't have ears. They don't make sounds anyway. But so what about chemical smell? They smell the females, and that's what they do. So the males detect the females by pheromones that she produces all over her back. This isn't the female, but all over her back, there are chemical cues that she emits. And it's not like a bug where there's or an insect, I should say, where they open a gland or something. It's just part of their skin lipids, just like we have. We have greases covering so we don't think of ourselves as being greasy, but we are. All terrestrial life is covered with waxes and greases, basically, to keep us from drying out. And so we can do kind of, this is just another cartoon of the kind of things you can see. We do a lot of our stuff right up here in the field. This is one of my former students, Mike Lemaster. He's a professor up at Western Oregon, and you can extract the pheromones off a living female right here in some solvents, and then you say, oh, that's bad, Bob, that's terrible, you do that stuff. This is like when you fill your lawnmower with gasoline. And if you're like me, you're always kind of slopping around and it spills on your hands and your hands turn a little bit gray. You wipe them on your pants and 5 minutes later, you're like, they're not gray now because the lipids came back. Well, it's the same thing with the snakes. And so then Mike's doing some fractionation here, or extractions, fractionation. And then this is the workhorse. This is some chemistry. We have sort of high-powered chemistry for this sort of low-powered kind of science here. We do gas chromatography, mass spectrometry, and then NMR experiments. So anyway, so we have that in our own labs, which is nice. So then I would say that we have robust courtship behavior. So show me somebody that can do this with a mouse or an eagle or I don't know, anything else. So there I am holding a female, and you can see these males. First of all, the female seems fine, and the males are just going up and courting, right in my hand. They couldn't care less. There's a female there. They're going to try to court her. You can see them crawling right up. And so they have very robust courtship. Pretty neat. This you can see also, that's out in the field. So this is right out in the dens. To make a really long story short, we were able to figure out, I guess I did this when I was a grad student and an early postdoc student. That was a long time ago. Some of you may not even have been born then. Whoops. And so go back. And so what it turned out is that the pheromone is this, it's the homologous series. It's all these peaks, and these peaks with numbers on them, that's the molecular weight of the peaks. And they're a series of saturated, which means it's straight chains like this, or monounsaturated. You've heard about polyunsaturated fats and monounsaturated and saturated fat stuff. This is a saturated fat, A. This is a mono unsaturated and what is called a methyl ketone. So this double bond O and a methyl group on the end here, that's the functional part of it. So there are these are kind of kissing cousin molecules, but because of that double bond, there's a kink in this molecule, and that's very important. And so that's what these are. These pairs like this, one is the saturated, one is the unsaturated. One is the saturated, the other is the unsaturated. And so the same thing like this. And so they're a series like this. So the other thing that I like to tell people, God, I cannot find my arrow here. Where is my arrow? There it is. I tell people early on in the insect literature, they would identify a compound as the pheromone in the, pine bark beetle or the cockroach or the silkworm moth. And all of those have come to be, they're not wrong, but they identified the most prevalent compound. Almost every pheromone system I can think of has multi-component units like this one. So this idea of, it's sort of like the one gene, one enzyme theory we used to have in biology, and that's kind of gone the way of all good things too. So this is very, and so this was the first pheromone that anyone ever identified in a reptile, even though reptiles are a huge class. So I was lucky, probably. I just fell into it. So this will show the pheromone bioasset. So again, I said, I'll let you guys decide. What this is if you go in the bathrooms, probably there at U of O or up in here at OSU, you can pull these brown paper towels out. This one, I've put the pheromone that we identified on that. This one is the hexane solvent. And what we did later is all the other compounds we found in there, we tested over here. And this is what you can see on the left side. And right here, that's the closest thing I can show you to a mating ball occurring on a paper towel. And so then people say, well, Bob, how come you don't have a model of a snake there, a nice rubber snake? And I said, because even though I really like working with these snakes up, they're not all that smart. I'm sorry to say. They don't have a frontal cortex, things like that. So they don't need a model. So they will try to mate with the paper towel. They'll pop their hemipenes out. If there's a little crease in it, they'll try to, do nasty things in there. So you don't need a model. It just goes to show, if it smells like a female, it must be a female. It sure doesn't look like a female, but it must be one because it smells like one. A mouse would never do that. Mammals don't do that. So that's another reason to try to work on these things. Mammal pheromones are very hard to figure out because mammals, like a mouse, they have pheromones. If pheromones don't work, they have auditory cues. They sing to each other. They have tactile cues. They learn. All of those. So one thing is knocked out, but all the other ones work. So it's very hard to figure out what they are. In our snakes, if the female doesn't produce the pheromone or the male doesn't detect it, nothing happens. They cannot reproduce. All right. Let me show you a few kind of fun things we've done with this. That's what this whole talk is going to be, kind of a grand tour, a whirlwind tour. So one thing was that we noticed right from the beginning is, just like in any other critters, you know, there's big females and smaller females. You know, there's like older ones and younger ones. Reptiles have what's called indeterminate growth. They grow till, as long as they're alive, they keep growing. But of course, once they get older, they're not, you don't see like 15-foot garter snakes around. They slow way down, but they are actually still growing. Unlike mammals that stop growing. In fact, you get to be my age and you start to shrink. I just went to the doctor and they chopped 1/4 of an inch off of me, or 3/4 of an inch. So what this shows is large females are preferred over smaller females. So you can see over here, and that's shown in the graph. These are the males, these bars. So males prefer to court larger females than smaller females. If we were live together, I'd say, who can think of why that is? The reason is larger females will have more babies. So if you were given your druthers, you would rather mate with a larger female because she could potentially have more offspring that you would be the father of, as compared to a smaller female. So then one of my grad students came up with the question, well, if they do that, you know, we know they're doing that there. Can they do that based solely on the chemical cues that are in those skin lipids? You follow me there? So it's back to like that picture I was showing. Do they just see they're bigger, or do they know from the chemical cues? So this was kind of a clever experiment that one of my grad students did. Here's the data I already showed you. Where's my arrow? Here. Here's large females. So males, here's the proportion of males courting. Large females really like to court males really like to court large females as opposed to small females. Here is males prefer large female skin lipids as opposed to smaller female skin lipids. So the same pattern is maintained. There's no snake there, just the lipids, just the pheromone cues. So this is a very busy slide, but I'll take you through it here. So here's on the right side, these right panels, these are those, remember the chromatogram I showed you when we identified the pheromone? Here's those methyl ketones again. And remember I used to have I shouldn't say, on the other one I showed you the numbers were the molecular weight. I should have said also that those molecular weight, those are very large numbers, so they mean that these signals are non-volatile. So that's another one. A lot of people know about insect pheromones where they're volatile. They're like a perfume. They spread, and everybody's heard the story about moths that can fly 1 kilometer upwind and find the female. These guys are virtually not volatile at all. So they have to touch the female, thus the chin rubbing and the tongue flicking. Okay, so that's what these are. And the dark ones here are remember I talked about the saturated methyl ketones, and these white ones are the unsaturated methyl, the one with the double bond, the methyl ketones. And then where this picture is, what this comes from is over here. And in this case, we're looking at the snout vent length of the female. How big is this female? So these are the small females. And then this shows how much methyl of the unsaturated methyl ketones they have. I'll show you in the picture in a minute. Here's B. This is a medium-sized female. Here she is over here. See how this chromatogram looks different from that? They're the exact same components, but they look different. So in this case, the small females have mostly saturated methyl ketones. See that? They're mostly black. The middle-sized one has, I would say, a mixture of both unsaturated and saturated. And if we go way up here, the largest females that the males like the most, they're down here. They're mostly unsaturated methyl ketones and very few saturated methyl ketones. Now, the thing I didn't tell you that I'll tell you now is the unsaturated methyl ketones are about an order of magnitude more attractive to the males. So if you gave them just unsaturated methyl ketones, they like those a lot. If you gave them saturated methyl ketones, they still like them, but not nearly as much as the unsaturated. So that's how the males can tell the difference from the females just by smelling her. All right, let's keep going. So then you can do all kinds of fun stuff here. Has anybody seen this one? This one, I know I'm talking to U of O, but I like to call these the OSU snakes. You know, they're black and orange right here, right? So these are red-spotted garter snakes, but luckily we have seeded them down in Lane County too. No, I shouldn't say it. They're all over Oregon. So you should see a lot of these in parks if you're walking your dogs. And this is the red-spotted garter snake. What we're talking about over here is the red-sided garter snake. I didn't give them the name, so, but you will see that these two are the same species, Thamnophis sertalis. See over here, Thamnophis sertalis, but they're a different subspecies, Parietalis and Consinus. And so these guys are actually the same species, but they don't live next to it. We don't have these here in Oregon, and we don't have our Oregon snakes in Manitoba. So we can look at, I wonder what the signaling is like there. Do the pheromones look the same? Here's another one. Now, this plains garter snake, you can see they're all kind of the same thing, stripy guys. This is Thamnophis radix, plains garter snake. They live in the same area as the red-sided garter snakes up in Manitoba. So those two live with each other. So wonder what their pheromones are going to look like. And if we're down here in Oregon, here's another new species. This northwestern garter snake is another one many of you might have seen, Thamnophis ordinoides. That one lives, as we say, sympatrically with this red-spotted garter. So they're right here in Oregon. So is there any confusion of all the signals and so forth? And so we can use our chemistry to do this kind of stuff. Forget this figure up on the top here. That's actually the data. What you can do is you can take all of this very complex data here, and then you can, it's a relatively complicated, it's pattern recognition. It's what's called a multi-response permutation procedure. At any rate, it's just looking for patterns. So does the pattern of one species look very much like the pattern of another species, or are they different? And in this case, you can put clouds around them. So up in Canada, the red-sided garter snake here, the black triangles, and the plains garter snake, the white triangle, can you see that? The clouds don't overlap. Their signals are different. So if you ever encounter a member of the other species, you don't make a mistake. They're both garter snakes, they're kissing cousins, but that's about it. They're just kissing because they don't make that mistake. Same thing in Oregon. So we have these two live sympatrically, but you can see the clouds of points on their pheromone systems, they do not overlap. So it's kind of cool how evolution has made it such that even using that chemistry, they've sorted it out. So you don't see hybrids of these things coming up and so forth because they don't make those mistakes because the signals are different. Okay, so I've shown you a little bit about this. So basically from one tongue flick, the male picks up these pheromones off the female's head with his tongue, and then he delivers it into his vomeronasal organ, which I told you I'll get to in a bit. So with one tongue flick, that's it. Snakes get a lot of information. I showed you they can tell the difference between males and females. I didn't show you this, but they can tell a female that's in the breeding season versus the non-breeding season. Their pheromones will look different. I did show you that they can tell the difference between a small female and a large female. And I didn't show you that they can even tell the difference between their snakes from their own den versus snakes from a different den. So there's a heck of a lot of information in just one tongue flick. So it's kind of cool. It makes them makes you appreciate them a little bit more. They're very sophisticated like that. Okay, here's another kind of alternative bioassay here. So we were up in the den there. You can see the rock behind these guys. These were 3 Cub Scouts or whatever they call them up in Canada. And they said, what are you guys doing up there? And we said, oh, we're doing this stuff we want. You know, we always explain it to the public. Oh, really? Well, let us give that a shot. And so here you go. And I like this kid in the middle here. He's like got that snake like right up his nose. And so these 3 boys said, sorry, this doesn't do anything for me. And I'm like, well, that's good because you're not a snake. And so you shouldn't be able to detect the snake pheromone. So that's really good. Alternative mechanism or method, but not a good one. So thanks, boys. Here's something I won't get a chance to tell about, but it was a really fun one. Up in Canada, the snakes used to get squished on the road because when they mate, I'll show you this in a minute too, but then they mate on the, after they mate for about 4 to 5 weeks, they all leave the den. And you would think if you hibernate, I guess I didn't tell you all this, those snakes up in Manitoba, they're the most northerly living reptiles in the Western Hemisphere, if not the whole world. We and the Swedish snake geeks kind of argue about who's got the most northerly snakes, and frankly, I'm not even really sure. It might be the Swedes, but it might be us too. And so they're very far north. They hibernate for 8 months of the year underground. If I were coming up after 8 months, first thing I'd want to do is eat a juicy frog, but they don't. They stop. They don't eat for another month so they can mate. So now they're going 9 months without food, and they're using a lot of energy, stored fat to mate and do all that stuff. All right. So then they have to leave after the mating. So then they leave and they disperse away from the den to all the compass points around the dens, and they can go up to 20 kilometers away, which is a long way to go on your belly in a short period of time. And then they eat fish and frogs and things like that. Well, in this particular dens we were working at, Highway 17 is like, it's like Highway 99 W or E. It's a 2-lane road, but way up there, that's like a major highway. And the snakes have to cross that, and the people aren't mean, but the snakes were getting really killed. So they were losing about 20,000 snakes a year getting squished by cars. And so the people up there said, yeah, somebody's got to do something about this. So they called the wildlife guys, and they said, the wildlife guys were like, what do you think about this? And so we were on this task force. Manitoba Hydro is the electric company. All their power is hydroelectric from dams and things. So they call the electric company Hydro. Everyone they don't say telephone poles, they say hydro poles because it's all hydroelectric. At any rate, it was incredible. So the electric company came in because they own the right of way. The gas company came and said, we can put pipes through the ground into the existing roadbed. Because at first we said, maybe we could just dig a tunnel. And they said, forget it. You can't wreck the roads up in Canada. It's like $1 million a kilometer or something like. When they get those roads and they're frost proof and stuff, they're never gonna break that road. It's like, you can't do that. That's like, you might as well you might as well want to rob the queen or something. Like, that's not going to happen. So they said, okay, but we can pull these pipes through and make a tunnel. And then they said, Mason, you and your geeky scientist friends, you're going to get the snakes to go through these tunnels. And I said, whoa, that tunnel is cold, dark, and like all things snakes do not want to do. They don't want to go in the cold, they don't want to go in the dark, and they don't want to go in a tunnel. And they said, well, what about all this pheromone stuff you talk about? You say they could follow trails, they could follow each other and stuff. Come on, let's let put your money where your mouth is. So I have to say, I'm sweating bullets. It's like, it works great in the lab, works in the experiments. I can write lots of papers about it. But anyway, long story short, we were able to get, unfortunately, all those dead guys on the road. We could extract a heck of a lot of lipids. We could salt it through the tunnels, and then we all got, and I mean, it was everywhere. Like, the citizens were out, all these the premier of the governor of the province was up there. It was a big deal. And I was like, oh my God, and I'm sweating bullets, like, oh, these snakes better do this stuff. It's like, I never tried this stuff before. They work like I said, they work in experiments and stuff. And sure enough, the first guy gets up there, he looks, takes a sniff, and boing, he goes right down the tunnel. And then they just started going crazy, just going through the tunnels. And it was, yay, everybody cheered, woo. And so now they've gone down. There's less than 1/10 of that. They lose like about 1,000 a year, and that's just where the tunnels don't exist. So that was pretty neat. So that was kind of fun too. So that got a lot of press all over the world because it was pretty cool though. What I liked more about that, not our part, was that everybody got together. It was incredible. Pretty neat. I'm not sure that would happen here, to tell you the truth. All right, let's look at some of the other stuff. So again, let's look at that tongue flicking and stuff. So in this case, that vomer nasal organ is way over here. So where's my arrow? Come on, arrow. Here it is. So they have 2 nostrils or nares right here. And here's their nasal canal, just like we have, just a nose with, sinuses and things. That's what they have. They can smell just like we can smell. But our smell is for volatile things. Think about, you go into grandma's house, she's made chocolate chip cookies. As soon as you open the door, woof, you smell that beautiful smell. That's a volatile odor. But snakes, and actually most other vertebrates except for people, are really good at smelling non-volatile things. So think about your dog. Why doesn't the dog go when it's the middle of the winter like this, it's raining and stuff? Do you have to go and sniff every telephone pole and mailbox in the whole neighborhood? Come on, just do your business. Let's get out of here. Well, they don't, as you know, because they are sniffing and getting a lot of information from the non-volatile cues. The best example are bloodhounds. And I always tell my students, I say, they always show these things in the movies. They're almost always wrong. They got a handkerchief or whatever. If anybody knows about these, when you get a bloodhound like we do in the Northwest here, people get lost in the woods all the time. What you really want to get is something close to your skin. So not to be indelicate, but you want to get underwear, socks, something like that. Not a sweater like I'm wearing. So that's useless. You want something that's touched a lot of my skin. Underwear. And then what does the bloodhound will sniff it? And does the bloodhound, here's a good question for you. See, I can't help myself, the professor. Does a bloodhound go through, I don't know if you can see, does it go through the woods like this with its nose up in the air? Or does it go through the woods like this with its nose on the ground? Well, you all know its nose is on the ground. It's not sniffing volatile things in the air. It's sniffing non-volatile cues on the ground. Like in this case, the person's skin lipids that it smelled in the underwear. Okay, snakes do the same thing. They live close to the ground. Why do they need to smell stuff that's up in the air? They don't. They want to smell stuff that's right in front of them. That's what they're trying to get. They're trying to catch worms. They're trying to catch frogs. They're not trying to catch a frog that's across the yard. They're trying to get the frog that's right in front of them. So that's what they do. So that's this vomeronasal, where is it? Here's our vomeronasal organ. And as you can see, it communicates with the roof of the mouth. So like in our soft palate, there's two ducts that come out. You can see two holes in the roof of their mouth. And so the tongue, where's my arrow again? The tongue comes out of the mouth, goes back in again, and it delivers chemical cues up into that vomeronasal organ. All right. So lots of critters have, and this is what it looks like. It's a beautiful thing on the right here. It's a mushroom-shaped thing. I cannot see my arrow to save my life. Where is my arrow? There it is. And so it's this mushroom-shaped thing. You can see that picture over there. And then it's got this, you see this big white? This is the vomeronasal lumen. So this is a fluid-filled space, and all the sensory neurons, just like in our nose, are all along here. See this all along the outer edge like this. This is where all the chemical receptors are and things like that. Same thing in the dog, okay? And so then there's, and as you can see, it communicates with the roof of the mouth. Okay, so what that means is this fluid that's in here can go both down onto the tongue and out into the environment, and comes back on the tongue with chemicals dissolved in it, and get introduced back up into this aqueous fluid. Okay, so that's how they can detect chemicals. This is just a cool picture to show that we got this. This is kind of neat stuff there. These beautiful cells. Here's the cell bodies, and here's the sensory dendrites here, and then the axons go this way. So these are this black out here is like the white here. Okay, so that's way out in here. All right, here's our molecule again. Here's something to tell you about. Here's a mystery now. Here's you can be our labs. Okay, this is all well and good, but remember I told you these are really non-volatile long chain fats. Remember I talked about saturated fats and things like these are fats. This is a lipid. This, you know how they say oil and water does not mix? These are oily molecules, oil, and that vomeronasal organ is filled with aqueous water, a watery fluid. So how do you get this wax, just like a candle wax? How do you get this waxy thing to dissolve in the water? Think about how you do that with beeswax. You're not going to do that. So that's a problem. So like, we know the snakes are doing it, so what the heck is the story there? And so here's the problem. Yes, as I said, the pheromones are insoluble, but they've got to go to this aqueous soluble organ here, and that's why there's a big red X. That's not going to happen. So how does that work? Well, I have to give the credit to one of my colleagues, Mimi Halpern, whose picture will come up in a bit. She's retired now, but she worked in the medical school in New York, downstate medical school. And she was an expert on the vomeronasal organ here. And she said, so we were working together on this, and she said, Bob, have you ever heard of the Harderian gland? I said, yeah, I've heard of it, but that's about it. And she goes, do you know that the Harderian gland right here, gosh, where is my arrow? I cannot see it. How come I keep losing it? Oh, there it is. Okay, this Harderian gland over here, I'll tell you about in a minute. It's this big thing. Look, there's a snake like a garter has a big eye. And this Harderian gland is a big, big gland. There's one on the other side of their head too. And then it goes into this lumen, this duct, and this duct has a blind, it doesn't communicate with the eye. A snake, all snakes have a capsule over their eye. So snakes don't have eyelids, if you look at that. Lizards have eyelids, snakes do not. So it's a blind-ended duct. But then can you see where it goes? It goes all the way, and the only thing it does, it butts up, it terminates in the vomeronasal organ. So is the fluid, so you can do experiments on this too. We now know the watery fluid that surrounds the vomeronasal organ does not come from the vomeronasal organ. It's not made there. It comes from the Harderian gland. Whoa, the plot thickens. Okay, so this is kind of cool. It was first described in 1694 by a guy, guess what? Jacob Harder, a German anatomist. Lots of things have been said, what it does: lubrication of the eye, thermoregulate, lipid production, immune function, pheromone production, porphyrin production, all these things that are just kind of one-offs. But all that work was conducted in mammals and birds. Why didn't anybody look at reptiles? Reptiles are the progenitors to birds. You guys probably all know about this. You probably had sessions in the museum. Dinosaurs are just living birds. That's really not true, but birds come from dinosaurs. The Harderian gland evolved before either of these groups. So what is the ancestral function? Don't look at it in birds and mammals. What is it doing in these reptiles? And this guy right here, it is arguably the last remaining large organ of widespread distribution among the vertebrates to which we cannot confidently ascribe a confirmed function. This was in 94. Let me tell you, it's not the case now because we've done this, but it was the case before we did this work. That's a big gland. Humans have this, if you're wondering. We have this gland. That's a very big gland. It's bigger than our salivary glands. So it's a big deal. Nobody knew what it was. All right. So what we're going on now is, and then I should probably cut us off on this. That we're talking about that Harderian gland. It produces fluid, but it also produces proteins that are in that fluid. And one of the proteins that we hypothesized is that it could be a pheromone binding protein. And that is just what it sounds like. You can get a pheromone, in this case, like a lipid pheromone. You bind it to a protein, and then it can travel through an aqueous environment. So the protein is able to dissolve, not dissolve, but basically able to exist in the aqueous environment. And it's got like a little kind of a cup that it's called a calyx, actually, the Greek word, and it's a little basket, and it can take that lipid pheromone and take it through that aqueous environment. Okay. And so that's a way. Let's see. I want to get to a stopping place here. The pheromone is insoluble in water, but the Harderian gland, and this just shows a picture of it there. The histol, it's a beautiful gland. Could be producing these pheromone binding proteins. This just shows you can inject ink into the Harderian glands behind the eye there, and within seconds, that black ink you can see coming out of the two ducts up in the roof of the mouth. This is the top of the mouth of the snake. He's up on, that's the top of his head there, and he's upside down, and so you can see the ink coming right out of those vomeronasal ducts. So that's the idea there, what I was just talking. So now there's a way for the pheromone to interact with Harderian gland pheromone binding protein and be able to be detected by the vomeronasal organ. So we found that was true. If you radiolabeled it with tritium, the more Harderian gland substance you had, where's my arrow again? The more substance you had, the more pheromone was able to be dissolved and detected. And so then the biggest thing is you can, this sounds mean, but you can actually do a surgery and you can remove the Harderian glands from the males. And if you do that, if you compare that, this is the how many males courted. So if you're intact, nothing's done to you. I don't know why one of these guys didn't do it, or two of these guys didn't do it, but mostly everybody courts the female, like this one, the sham one. They all court the female. But if you don't have your Harderian glands, you cannot detect that female. Okay, and so let's see. I don't want to do this. I want to show you one more thing. Come on. Why isn't it going? Come on. There, okay. Here's just one more, then I'm going to stop for questions. Okay. Are you going to go? Go. Ah, come on. Ah. Maybe not. Geez, I just had this working. Why isn't it working? Oh, buggers. All right, well, what this is, that's a little piece of worm. It's a little piece of rubber worm. And the cool thing is my my colleague, Mimi Halpern, remember I told you the prey pheromones? Garter snakes love to eat worms. She was able to identify what is the chemical substance in worms that drives garter snakes crazy. And what this movie would have shown, if I could get it to run, is that guy will do one tongue flick of this and then rawr, he like grabs it. Okay, I think I'll stop there. That's the compound. Yeah, I'll just stop there. So to make a long story short, we were able to find those pheromone binding proteins. We were able to identify what was going on in this long mystery organ that's even in humans. We were able to do some very cool molecular biology too. And so I don't have time to show you all that, but it's kind of a neat thing at the end. And I love to tell students that. So you can see this crazy professor and his students. We go out in the field, we're doing field work, we're in the lab, we're doing hormone work, we're doing chemistry with, sort of sophisticated chemical things, then chemical instrumentation like mass spec and NMR. And then we're doing kind of sophisticated molecular biology, doing genomics and a lot of transcriptomics. And so it's just super cool to be a biologist because, transcriptomics, stuff like that. When I was a grad student, no one even heard of that stuff. No one imagined it. And now the kind of experiments you can do these days are just unbelievable. All right, you know what? I think I'm going to stop there in case people have questions. So Lauren, let me see. I can't see the stop share button. I'm covered up with our pictures. There we go. All right, let me get out of there. There we go.
All right. Thank you so much, Bob. We have had a few questions come in, so we'll go ahead and jump right in.
All right, yeah, jump right in. Yeah, get them.
Someone is asking whether I believe they're the red-spotted garters, the ones that we have here in Oregon. Yeah. They do similar mating congregations.
Yes, yeah. Okay, keep going.
And if so, if you have any recommendations on good spots to go and find.
Oh, okay. Yeah, so the question, it's a great one. So this is, remember I said the exception illuminates the rule. Actually, all snakes reproduce the same way, except you might just have a male and a female, and the male will chin rub and tongue flick the female, but basically, not basic, it's exactly the same, except there's only two of them. So our red-spotted garter snakes do that. That is a tricky one. Our guys do hibernate. They're coming out right now, not now when it's rainy, but the next few days when it's warm enough and when it's sunny. It doesn't have to be bright sun, but just sunny. So it doesn't have to be hot. If the sun is out, they're basically black bodies. They absorb that heat. You can see, what I always tell people, if you ever see more than two snakes at the same time in Oregon, it's almost a sure thing that one's a male and one's a female, and then just follow them and watch them. And then eventually, usually the female is running away and the male is chasing. And then a lot of times, most of the time, even for these red spotteds, you'll see maybe 3 or 4 or 5 males. And again, that big female, and you'll be able to see the whole thing. It'll be just like this. Yeah. Where to find them? That's a tough question. They're all over the place, but they have their own little dens, but here in Oregon, it's just like a hole, like a gopher hole or something. So there aren't any den sites like that up in Manitoba.
So someone on Facebook is asking if the snakes prefer mating with snakes from their same den or from different dens. And do they always go back to the same den, or do they hibernate in different dens?
Beautiful question. These snakes are, like all reptiles, are very long lived. They can live, we don't actually know their limit, but 25, 30, 35 years. They're extremely faithful to their dens. So we've done lots of mark recapture. If they leave a den for 20 kilometers, they come back to the exact same den. And so I think so that's neat. The interesting part is the babies. The babies are born out in the wild and don't come back to the den that first year. And they are the colonizers. So they don't follow mom, or the mom doesn't take care of them anyway. Was the first part, Lauren, I'm sorry, was that, do they like the ones from their den or not?
Yeah, because you had said they can tell a female who is or isn't from their den. Do they have a preference?
They can, and it's actually, I think it's very interesting. That questioner might too. They actually prefer females from their own dens. And so most of the times when you get that, you have, and Charles Darwin is the one that described it, you want to have inbreeding avoidance. But, the chances of you breeding with your brother or sister out of 75,000 snakes is, it's probably vanishingly small. You can do the statistics on it. And so lots of people have talked about that, inbreeding avoidance, things like that. That's easy to tell, because usually if you have inbreeding, you have dead offspring. And so that's an easy thing to detect. But Charles Darwin in the same paper wrote about outbreeding avoidance. And so that's much harder to identify. But in that case, there could be characteristics or traits associated with that particular den that you want to maintain those traits. So it's a very interesting one and what we did not predict at all, but they have a very strong affiliation for their own females from their own. And we think that is outbreeding avoidance. Great question.
So we have time for a couple more.
All right, I'll be quick.
Someone is saying from the tunnel story, it sounds as though males are receptive to the pheromones even outside of the breeding season. Is that correct?
Yes and no. It's no. So let me say that. So the big one, no. So at the end of the breeding season, when they're leaving, so a female will leave, like 2 or 3 days after she emerges from the den. So she's very attractive, things like that. And then when lots of females are leaving, the females can remate. Once that plug dissolves in about 36 to 48 hours, she becomes sexually attractive again, which is these days you get through. But the pheromones are attractive again, and the plug is not there. So the female can choose to remate again, which is really cool because they can store sperm and they have multiple paternity within litters. Again, something we can do these days with modern molecular biology, which is really cool. And so it can behoove a male to follow the trails for females. It's a little bit complex. In the bottom of the den, a very large female can mate with a very small male, which is not something you would predict. You would usually predict size assortative mating. In other words, big females mate with big males and little females mate with little males. And that's a truism throughout the vertebrates. Except in these snakes, it isn't. It's almost like chance in the den, because it almost is. The female, I said, she determines when to mate, but she lifts her tail, but it seems like it's impossible for her to pick the male in the den. When she leaves the den, many of them will stop, let the plug dissolve, and then remate. And when they remate up outside of the den or on the way to the feeding grounds, there are much smaller mating balls, and their size assortative mating is very strong. So a large female will choose and can choose at that point a large male to mate with. So it's very, very strong size assortative mating. So that's a really long-winded answer to say that even when they're leaving the den, They can still be following the pheromone. And then when they come back to the den in the fall, it's really cool because they become attractive again in the fall before they go into hibernation, and they mate, even though they're not going to fertilize the eggs or anything. And so again, part of that multiple paternity. So both times when they're leaving and coming back, they do follow trails.
The follow-up to that was, why do the females go through the tunnels?
That's a good question. What I think is going on is, well, part of the thing is I lied a little bit about that. Not lied. I left, like all good stories, I left out a little bit. We also put drift fences down there so that they follow the fence and then they have to go through the tunnel. But when they're coming back in the fall, they do just go through the tunnels. And that's because when they get close to the den, the snakes seem to be trailing one another, males trailing males and females, whatever. They're just trying to find the den. They're not worried about, even though they become attractive again, they're not too worried about mating. It, like I said, it can happen in the fall, but not a lot. And so what they do there is they just seem to be saying, like, I got to find this den, which you can imagine if you're looking on your belly and it's as big as a living room, that's not really all that big. And so what they do is they follow trails. And if you think about it, the trails are all like the spokes of a wheel, a bicycle wheel. The hub is the den. And then the spokes are these chemical trails from all snakes coming in from all different angles. So if you follow those trails, you can literally, and we've put radios in them, they circle into the den and then boom, they find it. So they do follow, both the males and the females will follow each other. But that's a very good question. There's no reason in the springtime, the females they have no reason to follow each other or males or whatever. Great.
So we'll wrap it up with one final question.
Okay.
Someone is wondering if you have any advice for how a kid who's 8 might find mentors for herpetology.
Oh boy. Well, I can be somewhat self-serving a little bit, but it's not me. Somebody at that age is perfect. A woman named Cy Montgomery, who's an award-winning author, nature writer, wrote a book about, sort of me and my work. Anyway, it's called The Snake Scientist, and it's a great book. I guarantee it's in the libraries there. I said that's probably going on, that might be 20 years old. And I still get emails from all over the world, literally, about that. It's a great book. I don't get any royalties off it. Cy wrote it. But it's a really good book, and it's for kids. It's by Houghton Mifflin. Lots of schools have it and so forth. To do that, the bigger one, I don't really know. There may be actually through the museum there, contacts and stuff. There are a lot of what we call amateur herpetologists who probably know more about real herpetology in our areas, in any area in the country, than the eggheads like me. I'm pretty good at Oregon and stuff, but I'm a Connecticut kid. I grew up in New England, and I know a heck of a lot about Manitoba's wildlife, but I'm often, in the springtime, I'm up there. And so the local herp clubs and things like that are great ones. A lot of times they have jamborees or whatever where they go out, and they are a blast. And so that I would urge, I don't know if you said that it was a mom or a dad, but I would urge that parent to, kind of, if you poke around a little bit, I'm sure you'll be able to find something there, one of those groups. And they're really, really good. They love to get other people hooked on there. They're like bird nerds, so they love to get people hooked on it too. So we've had lots of stuff like that up here too. So we love to have the kids come along. They're great, and they're closer to the ground, so they can catch the snakes better than we can. All right.
Well, thank you so much. I want to thank everyone who tuned in. You might have noticed a little something over my shoulder here. This is just a little tiny sneak peek of one of the photographs that is in the photo arc exhibit that I mentioned is opening on Friday for members and on Saturday for the public. So come on by, check it out at the museum. And we have this, I believe, rhinoceros snake right here, but tons of other really amazing photographs. So come on down to the museum. Thank you again so much, Bob, and have a great night, everybody.
All right. Thank you, Lauren.